So welcome back. This is E340 542. This is lecture 6 because we missed last week. And we're going to be talking a little bit about thinking about models, and we'll talk about using the event handlers in Antimony, and then we'll also talk about a few motifs basically from the homework. And as always, this is live streamed and recorded. And the chapter reference is really chapter 13 on stability theory. The textbook doesn't really cover events very much. They're not very complicated. So we're going to start out by Hayden talking a little bit about a project that he's involved with, which would be nice to be able to generate for the class as the project. In old years, we published the projects in the form of notebooks on NanoHub, which is a Purdue-run server system. But NanoHub is getting a little bit old, and so it makes more sense to publish code in the form of Jupyter notebooks through Colab, and then Hayden will talk a little bit about how to support that. And then I'll remind you again about that variable stoichiometry, and we'll talk a little bit about how to describe what you see in a time series, how to analyze questions. And then we will quickly cover events in Antimony, and then we'll jump into some motif analysis. And in particular, Herbert has some code in chapter 13 that we'll use, and that's in one of the notebooks that I shared, but I saw in the homework you handed in that you had used it as well, so you've got that working. So Hayden, why don't I turn it over to you to start with, and you can talk a little bit about what you're trying to do and how we could potentially distribute a small project from the class.
Sure. So, Jen Shao, I don't know if we've officially met. So I'm Hayden Fennell. I'm a postdoc in James's group. And I've been working over the last couple of months on deploying a modeling course ebook website for another institution at Purdue that we work with called Embryo. Go ahead and share what we've got for that so far. We got the prototype hosted a couple of weeks ago. There's going to be some ongoing changes to this over the next couple of months. The idea behind this is that it's basically it's sort of two things. It's a course on how to do biological modeling kind of from the ground up. So it teaches you everything that you need to know about fundamentals of Python in order to work through the examples in the course, and then it'll go over necessary biological background, modeling workflow practices, a number of different things. And in addition to being a course, it's also going to be kind of a repository for a number of different example projects that we have that people can work on kind of once they've completed the modules, and the modules we'll be adding over the next few weeks. Right now we do have some example pages to look through. So let me put this link in the chat, actually. So here's a link to the current build of the site. Again, this will be changing a good bit over the next few weeks, but there are already some existing projects on the page which you can take a look at as examples. And the key here is that these are all pages that are on the site right now have all been built from Jupyter notebooks, which I don't know if I think have you been using Colab, James, for class so far? Yeah, so each of these has a corresponding notebook version James just pointed out that we don't need the apostrophe, which is true. I think that was subconscious autocorrect. But so you'll see that on the page here in static form, we have some background on ODEs and PDEs, finite difference method, number of different things, mathematical background. I don't want to go into too much detail here. This is just a quick overview. As well as code on how to actually run some of these examples and get output. And again, okay. Having issue with just the link to this notebook for some reason, wants to send me to text output. I don't know why that is. I need to figure that out, but the links on the other pages work. We'll take a look at this morphogen gradients page. This is based on a paper that was published in Developmental Cell, where they took the results of the paper and then built a Python model around it. So you see you can embed all sorts of images. And then here they have description of the model, results, and then the code that actually produces output relevant to the model. Okay, I have to open it. Should be a way just to link straight to the opened notebook. I don't know why it's suddenly not doing that, but yeah. So here we have the notebook version. So this will be fully interactive, so people can actually work through the example themselves and manipulate the code for different types of output. So I think that one of the reasons James wanted me to present this briefly today is that this site that we're building is actually a good avenue for publishing notebooks that people build as projects for this course and for other courses that we do. I think, James, you had a specific example in mind of something that could be built into an educational notebook that we could then convert into a page on this website, and obviously you would get full credit for that on the page.
Well,
Sorry?
I thought if you were interested in that P53 example, that's a nice didactic example, and the frequency locking is interesting, and the phase diagram is interesting. And so if we could, for the class, develop a little notebook version of that, then that would be a nice add-on, because there isn't a discussion of frequency locking or period doubling in this textbook so far. And so it would cover material that would be very relevant. And it sounded like it was something you were interested in because you'd worked on frequency locking in other contexts. And so if that were the case, I just thought that this might be a way. We don't have to put it up here. You know, it could be a collab notebook. But I thought this would actually make it, whatever work you do, less transitory, because it'll actually get published.
Yeah, and in terms of formatting it for the website, we have a pipeline set up so that if it's built as a Jupyter notebook, it's fairly easy to convert and put on the page. I could take care of that part of it, as long as the notebook is built and ready to go. So you wouldn't have to worry about formatting it for the web page or anything like that. That process is fairly straightforward.
I don't know what you think about that, but I thought when Hayden presented this last week, I thought it might be of interest to you. That's a possibility.
Sounds like a good project, yeah.
I mean, whatever the project is, it's still, I mean, we could do a different project, but it just, that this is a way of taking it. Again, I always worry when people write term papers or do projects and they disappear. And as a student, you do a lot of work on these things, and sometimes you'd rather that they disappear because you're not maybe happy with it, or you didn't spend so much time. Not you personally, but some people don't. But a lot of times people put a lot of effort into them and they come up with really great results. And it's a pity not to have those be visible to the public, so that you can point to it. I remember a few years ago, we had when this was a big class, we actually had two students who got jobs. Was they put in their resume a pointer to their class project, it was up on NanoHub. And potential employers liked it well enough that they actually gave them jobs based on this. So I think, I mean, you're in an academic track, but it's still potentially an interesting thing to do. It's a good thing to have. Okay, so I just thank you, Hayden. I just wanted to bring this up and put it out there as a potential option. So I wanted to come back. There are a couple of things that I was going to review. Again, we don't use this variable stoichiometry that often, but it is possible in Antimony with that annoyance factor that the stoichiometric variables need to have different names. And so then you have to slave them to a master when you're going to change things. But it is possible to do that. And so if you want to change the exponent, the only time you really want to do that is when you're changing the exponent in the Hill function. We typically will use Hill exponents of 4 or 8. Sometimes it's interesting, though, to see what happens when you change the Hill function, the exponent from 2 to 4 to 8 to 16. If you just have the exponent in the rate law, of course, that you can change by hand. It doesn't do any harm. But if you actually want to respect the stoichiometry, then you have to be able to change the stoichiometric coefficients too. So that's why we need that. Then we had done the analysis of one of the problems that was then in the homework, which was that autocatalytic single species autocatalysis. And we looked at the time series. We saw the bistability. We solved for the steady states as a function of initial concentration, and then we did simple bifurcation scans for the parameters in the simulation. And so I want to jump now to talking a little bit about thinking about models. And how bad is the background noise? There's somebody hammering in my background. Is it very distracting or is it tolerant?
We don't hear any.
Sorry?
I don't hear the background noise.
Okay, good. In that case, I'm going to just continue. If it becomes distracting to you, I'll go and ask them to stop hammering. But they're doing some, I'm at home and they're doing some repairs on the house. So I've got some hammering in the background. So typically when we're going to design a network or analyze the network, we want it to do something. And there are a lot of different things that it might do, but we often will ask for it to turn on when a signal is, for example, between 2 levels. So for a low input, we want it to be off. For an intermediate input, it's on, and then for a high input, it's off again. We might want to go from something that's more or less continuous in time to something that is variable in time. So we turn something on, it generates a pulse, and then stays off after that. Or we might do the other direction, which is we have a pulse as a signal, and we want that to switch a circuit from low to high, or vice versa. And so motif analysis is essentially trying to ask the question, what kinds of feedback circuits will generate the best responses that we want? Often we think about steady state responses, fixed points, but time dependence is interesting as well. And the motifs that were in that homework problem are ones that you run into biologically quite a bit. And it's really maybe not appropriate to say that a biological motif has a function, because these aren't engineered systems, but they do have behaviors, and we can understand those behaviors. I'm not going to blur over the background. It's annoying to me to see that. Thought it would work, but it doesn't. So another question that we run into biologically a lot in and this is becoming less true, although it's still true to a substantial extent, is that biologically it's very hard to quantify molecular species. Normally, we can't measure the exact concentrations of a molecular species. And often we don't really know what those exact concentrations would mean in any case. And so instead, what we do is we do what are called perturbation experiments or differential experiments, where we will do an experiment, get the level of something, essentially in arbitrary units. Then we will make a change to the system and ask the question, did that level increase, decrease? At least at a semi-quantitative level, we'd say, did it increase a lot? Did it decrease a lot or a little? If we can quantify not the absolute amount, but at least we can get ratios, we'll often express our results in terms of what are called fold changes. The amount of this molecule went up by a factor of 3, went down by a factor of 5. And that differential, thinking about differential measurements is fundamental to the way biological experiments are done. Physicists and engineers tend to think about absolute measurements more. But biologically, it's often not possible to make absolute measurements. And as I say, very often it's not really meaningful to think about absolute concentrations. Having the concentration double does something. Having the level be 0.1 nanomolar doesn't really mean anything. And so we often will do differential measurements, and even very simple biological circuits can have non-trivial differential results. And in previous incarnations of the class, we spent an enormous amount of time on the 3-element feed-forward network. I find that network, rather, analyzing it fully, it takes a surprisingly large amount of time. Herbert goes through it in some detail. And it's not really that exciting, although you can do things like pulse generation with it, so it's not trivial. And so what we have here is we have molecular species P1, and it does something to P2. It either activates, increases P2, or inhibits P2. P2 activates or inhibits P3, and P1 also directly activates or inhibits P3. Now, if everything is an activator, then when we increase P1, we're going to increase P3. If, on the other hand, we have P1, for example, inhibiting P2, and P2 activating P3, and P1 activating P3, we have what's called an incoherent feed-forward network. The direct signal, increasing P1, wants to increase P3. The indirect signal, P1, decreases P2, and that decreases the promoting signal from P2 to P3. So increasing P1 increases P3 directly and decreases P3 indirectly. And the net effect is going to depend on the strength of those signals. We can't determine whether the net effect is positive or negative by looking at the signal, at the network by itself. The other thing is that each one of these steps has a delay associated with it. And so if we have P1 activating P3 directly and P1 inhibiting P3 through P2, when you increase P1, what you'll see is P3 will come up and then it'll come down again. And so now you have non-trivial temporal response. That's how you can get things like pulse generators or filters with even this simple system. And so it's worth beginning to think about that kind of circuit as you look at real biological circuits. When you look at the actual circuits for gene regulatory or signaling networks, they're often pretty complicated. But this kind of feed-forward element is pretty common. And one might ask, well, why not just have P1 directly modulate P3? Why have two pathways for it? So that's something you can think about. And so this comes back to sort of a general question, which is what kinds of questions we can ask. And we think of a model as essentially a model structure, the kind of diagram that we just drew, and possibly the functional forms of the rate laws. And then there are going to be a set of parameters that could be the rate constants, the Michaelis constants, the Hill exponents, and so on. And that model structure plus those parameters and also the initial conditions then will give rise to some time series, some set of model outputs. And in our case, the output of all of our simulations is going to be a time series. It'll be a scalar value in time. And there aren't really that many kinds of questions we can ask about those time series. And so we can actually go through and systematically characterize the kinds of questions we can ask about a time series. And the reason I'm mentioning this is that pretty commonly, both as a research question and in a homework problem, I'll say, what do you see when you do something? And so this is addressing the kinds of questions that you can ask looking at a time series. And typically we'll ask those questions not only about some time series for some set of parameters, but also how that time series will change depending on the initial conditions of the variables, the values of the parameters, and potentially also on the model structure, although we tend not to do perturbations on the model structure so much in this class. Now we have to think about when it's appropriate to ask this kind of question. So the first thing we can ask are purely qualitative questions. And we can often get meaningful qualitative information from a time series, even if we can't calibrate the time series. And there's also the fact that typically our simulations involve a lot of parameters that we can't pin down very well. And so the numerical values, again, are going to be hard, but the qualitative behaviors should agree. If the model is going to be useful, it better have the right qualitative behaviors. If the qualitative behaviors are wrong, then certainly we don't have anything useful coming out of our model. And really, for a single value time series, there aren't that many things it can do. It can reach a steady state. It can oscillate periodically in time. We could have chaotic time series, or it can diverge. That's it. And so there really are only 4 qualitative responses we can have for a single time series. Now, if we have multiple time series, we can describe the relationships between them, and then we can get more cases. But we can certainly ask the question, if we have a given model structure and a given set of parameters, a given set of initial conditions and boundary conditions, does the time series reach a steady state? Does it oscillate periodically? Is it chaotic, or does it diverge? Now, biologically, we can never have infinite values of a concentration or a number of cells, and therefore divergence ultimately means something is wrong. The model is going to fail. But over a reasonable amount of times, divergence can be a sensible thing to get. But something else has to intervene to cut off the divergence. The next thing that we can ask, and you're a physicist really, so phase diagrams are familiar to you, is how do those behaviors change when you change parameters? We increase the value of a particular parameter. We go from a steady state to an oscillating state, bifurcation. We increase the value of a parameter. We go from an oscillating state to a chaotic state. Another bifurcation. We move a parameter and we change from one steady state to another. That's also possible. Or we go from a steady state or an oscillating solution to a divergent solution. Biologically, if we see divergence and it really is divergence, it's going to mean our organism is going to die. So that might be something we care about. And so we want to know how many different states we have of each kind and where their boundaries are in our parameter space. So we want a phase diagram. And in particular, when we make changes to a system, we want to know either how do we drive the system across a boundary. For example, if we think about cancer, in cancer, we have cells that are going through a cell cycle. And so we have oscillations in the cells, periodic oscillations in the cells. And if we want to stop the cell cycle, we might want to have some chemical manipulation or other manipulation of the system that turns the cell cycle oscillator off, moves us from an oscillating to a steady state situation. Alternatively, might want to have something that kills the cell, in which case we might want to go from an oscillating to a divergent solution. And so again, you're familiar with phase diagrams, but in general, the topology of a phase diagram, the domain structure of the phase diagram is pretty robust. And again, it's something that we would want our simulations or our models to replicate correctly. If there is a steady state region followed by an oscillating region, followed by a chaotic region, we'd like to have that sequence of regions be right, even if the exact boundary positions are not quite the same in the experiment and in the model. Now, this is a fantasy. It's not really quite what happens. But if I imagine a cardiomyocyte, so a muscle cell in the heart, those cells under normal concentrations of sodium and potassium contract spontaneously. They oscillate spontaneously. If you put them in an environment they don't like, they'll stop oscillating. It's a steady state solution. If you push them too hard, you can get chaotic oscillations, which is usually a bad thing for the heart. And if you really push them too hard, they'll oscillate until they die, and you get divergent solution. And so we can imagine for a particular system drawing a set of boundaries of this kind. And already in the homework, you drew a flow diagram, and one can think about relating that flow diagram to a bifurcation diagram. We did the 1D bifurcations in class 2 weeks ago. Here we're looking for two-dimensional bifurcation. So that's what we can do with qualitative observations. We have what I call semi-quantitative questions. And semi-quantitative questions typically describe relationships. And again, this is something that biologists do all the time because, again, it's hard for them to experimentally give absolute numerical values for things. And so we typically are looking for relationships between values or trends. The simplest one typically is, does A or B increase or decrease in time? And does something happen before or after something else? And then if we want to make a stimulus response question, if I increase A, does that increase or decrease B? Or potentially, if I increase A, does that speed up or slow down change of B? And you can certainly come up with more semi-quantitative questions. But the answer for each one of these questions essentially is going to be a yes or a no. It's a Boolean output. Now, you could make it a little bit more graded. You could say it increases it a lot or a little. But typically, these are Booleans. And so those yes-no sorts of questions are more robust to experimental noise, to errors in measurement, and so on. And again, if we don't replicate the semi-quantitative results, we're certainly not going to be able to replicate quantitative results. Another kind of question would be, for example, about steady states or long time behaviors. Is the value of A 0 at long times? Is it bounded at long times, or is it infinite? Does it diverge? And again, you can build phase diagrams related to semi-quantitative questions. If I imagine that I have a tumor, that tumor is either going to grow or shrink. And I can also look at the cells around the tumor, the normal cells, the stromal cells around the tumor, and I can ask, are they surviving or are they dying? And now I can think about treating the cancer with a chemotherapy. Chemotherapies, classic chemotherapies, are what are called cytotoxics. They kill cells that are dividing. So if I give a plenty of cytotoxic chemotherapeutic, I'm going to expect that the tumor will shrink. The problem is that if I give too much of that cytotoxic, the normal cells are going to start dying too, because it's toxic to normal cells as well as to cancer cells, just a little bit more toxic to cancer cells. Now, another kind of anti-cancer drug is one that restricts blood vessel growth. Cancers partly grow by inducing the formation of new blood vessels. And so I could imagine that as I change the concentration of a second drug, that there's a boundary between tumor growth and shrinkage as well in that direction. And as it happens, I also know that if I give out too much anti-angiogenic drug, that's also toxic to cells. And so in this case, I've got sort of a fantasy, but if I give too much chemotherapy, my normal cells are going to die. If I give too much anti-angiogenic, my normal cells will die too. And so that diagram on the right is actually pretty reasonable. The one on the left I made up because I wanted a particular result. But if I now put these two diagrams on top of each other, I'm going to get a region in which the cancer cells die and the normal cells don't. And that's the region in which I might expect therapy to work. And so I'll say, if the chemotherapy is too big, it doesn't work because I'm killing the normal cells. If the chemotherapy is too small, it doesn't work because the tumor cells are growing anyway. And if I look at this, there is no concentration. I've designed this, so there is no concentration at which, if I only have the chemotherapy, things work well. Actually, I was a little careless. I think there's a small region. If I now include the anti-angiogenic, I have a broader spectrum of region in which I can have the tumor suppressed and the tumor suppressed without killing the cells around it. And so the shaded region then is a prediction of combination therapy efficacy. And I said it wrong. If you use the anti-angiogenic by itself, there's no concentration at which it works. If I use the chemotherapy by itself, there's a small region in which it works. If I look up around here where it says normal cells okay, there's a fairly broad region. If I use the anti-angiogenic at the right concentration, there's a fairly broad region of chemos that are going to get rid of the tumor without killing the host. And that's usually what you want for a successful tumor therapy. And then you could ask other questions. Here we're imagining we're trying to get rid of the tumor, but the anti-angiogenic drug might it acts on the blood vessels. So you could ask what it does to blood vessel density. And we could ask about things like delivery vehicles. These days, a lot, because chemotherapeutics are quite toxic, they're often conjugated to antibodies or to liposomes that primarily target the tumor. So you get less of the toxic chemical in the normal tissue and more in the tumor. That can help as well. And so again, this is a fantasy, just as an illustration of the kind of questions that one can ask, depending on the particular biological problem you're working on. And of course, sometimes you can actually measure numerical values of concentration. And sometimes those are important. So you might want to know, what is the level of a molecule at a given time? Is the amount bigger or smaller than some reference amount at a particular time? When does an event happen? Not only does it happen earlier or later than something else, or faster and slower than something else. How frequently does an event happen? That's one of the few things quantitatively that's pretty easy to measure. If you want to know how frequently does a cell divide, that can be quantified. The levels of things are hard, but the time at which something happens is easier to measure. You can't get it exactly, but you can get it pretty well. And so that's just a general discussion of some of the kinds of quantitative questions that we could ask. Is concentration of A greater than or less than B? Is the rate of increase of A greater than or less than B? And of course, ultimately, if we're doing dependence on a parameter, you might ask what the specific functional relationship is. Now, these are typically questions that are very useful. How much of a drug reaches our target? How much ethanol does our reactor produce per unit of glucose? But those are also the kinds of quantitative questions are the ones that our models are most likely to get wrong. And so very often we'll be happy if we can reproduce the qualitative and semi-quantitative behaviors that are observed in experiment, and not worry so much about calibrating the model exactly quantitatively. So having said that, I wanted to come back to a couple of the problems in the homework. These were all designed to be slightly trick questions. And I thought maybe we could discuss them. Why don't we come back to the very first one, which was, what's the population of humans on the Earth as a function of time? And at some level, we know what's happened in the past, but we can make some statements about what's possible in the future. And so historically, human population has gone up. There may have been some plagues and periods of ice ages where the net population on the Earth went down a little bit. But for the past million years or so, human population has increased initially quite slowly and then more rapidly in the past couple of hundred years. But if we look forward in time, or even currently in time, what are the possible things that could happen going forward to the human population? Do you want to think about that a little bit together as a possibility?
The parameters that will affect population.
I'm sorry about that. I
didn't catch your question actually.
So the question was the homework problem, which was, what's going to happen to the population of humans on Earth as a function of time? And I think the answer you provided was that it keeps increasing. And
Yeah,
you
keep
said,
increasing, but there's a limit, I think.
right, there has to be a limit of some kind.
Yeah.
So one thing that could happen is it saturates and reaches a steady state.
Yes.
Another thing is that it overshoots the carrying capacity and then comes down again. And it could oscillate. Or you could overshoot so much that you run out of resources and you go extinct. So there are at least three possibilities, one of which is You go up, you saturate, and you reach a steady state, modulo noise. One of which is you come up, you come back down, and you oscillate. You could come up, overshoot, and come back and reach a steady state too. And a third one is you overshoot so much that something bad happens and you go to zero. Because once you have population of zero, it's not coming back.
I assume that the resources will reproduce and it's roughly a constant. So the population would saturate eventually.
I mean, those are all reasonable things, but
Yes.
But the point of the question is to think about the possibilities.
Yes.
It's not.
Mm-hmm.
I mean, if you assume there are unlimited resources, then you expect the population to keep growing. That's reasonable.
Yeah.
But biologically, those divergence never is possible. There's always some kind of saturation or limitation.
Mm-hmm.
Now, the second one is the rate of chemical reaction of A goes to B as a function of the concentration of A. Now, we talked about mass action, and we said that the simplest rule is that the rate of reaction would be proportional to the amount of A.
Mm-hmm.
And that's certainly possible. And if A is an independent reaction, for example, a decay reaction, then the rate of reaction is going to be a linear function of A no matter what the concentration is. So maybe that one is not so exciting. Although we can think of, we'll come back to the actually it's not, I don't have a bullet point in front of it. I managed to merge two together, but let's look at the next one. So what about the rate of chemical reaction A goes to B as a function of the temperature? I think in your homework you said, well, you expect that the rate of reaction will increase as the temperature goes up.
Yeah.
And that makes sense if it's especially if it's an endothermic reaction, then it's an activated reaction, and therefore the rate of reaction will go up a lot fast when you increase the temperature. On the other hand, the rate of back reaction will also go up. All right, the rate of reaction of B goes to A will go up. And so the net rate of A going to B, at least for low temperatures, will probably increase. But at high temperatures, maybe the back reaction is faster. You don't know. The other thing is that if the temperature is too high, A will break down. If A is a biological molecule and make the temperature too high, A will denature. And so at very high temperatures, basically you kill it. So there has to be some temperature at which the rate of reaction goes down again. Can't go to infinity. And so, again, I'm not trying to be critical of what you said in your homework. I'm just saying that these problems were designed to not have clear answers. They seem very simple, but actually, for some of them, the answer is we don't know, or it depends.
I thought this problem was meant to ask me about the general rules.
Right. It's trying they're just trying to encourage you to challenge your assumptions a little bit.
Mm-hmm.
And I'm not trying to be difficult. I think your answers were fine, but I wanted to come back to them because they do illustrate some of the complexities of trying to build models.
Yes.
Third one, which is run into that one fourth one, which is run into that third one we just looked at, is the rate of chemical reaction of 3A goes to B as a function of the concentration of A. And in this case, again, we would write mass action. And so we'd write the rate is K times A cubed. And for small concentrations of A, that's reasonable. But when we have high concentrations of A, we have things like steric repulsion. We can't squeeze that much A into one volume. And so almost always, when we have high concentrations of molecular species, the rate of reaction is slower than mass action would predict. again, there's a saturation. The speed of reaction can't go to infinity. And so for small concentrations, the rate of reaction will go as A cubed, but then it's large concentrations, there'll be a saturation. We don't expect the reaction to stop. So it's not that it's going to go, the rate of reaction will go to 0, but we do expect it to saturate. Now, the next one, if I remember, and you answered, that one did trigger some thoughts about that. The rate of glucose absorption by E. coli is a function of external glucose concentration. It really would be helpful for modeling biology if there were a simple rule that said as you increase the glucose concentration outside a cell, the amount of glucose taken up is this fraction of glucose. And in our models, we often use that. We say you take up 10% of the glucose or something like that, or we'll say it's a Hill function. But in reality, what's going to happen is actually pretty complicated. If the concentration of glucose out the cell is very, very low, the transporters may not work, and the cell may not be able to take up the glucose at all. Then there's a pretty big range of glucose concentrations at which the rate of glucose uptake is more or less proportional to the amount of glucose outside the cell. As the concentration of glucose gets to be very high, and you pointed this out, we expect there to be a saturation where the transporters are saturated. You can't take up any more glucose. And so the rate of uptake will saturate. And finally, what happens if you put a typical cell into honey?
It will die.
It'll die. So again, if the concentration of glucose outside the cell is too big, the cell's going to die. So then the uptake rate will go to 0. Or maybe it just doesn't make sense to say that there's an uptake rate. And certainly as the cell gets really sick, as osmotic pressure is drying the cell out because of the glucose in the environment, then the uptake rate will go down. And so there's going to be some turn off. And I think you talked about that as well in your response. So it's a little bit like the argument about the population of the Earth. We have to take into account the idea of saturation. And it's not wrong to say that the population of humans will go up as long as you're specific. And you were specific. You said if there is no resource limitation, then the population of humans will go up. And so provided that you specify the domain over which your answer works, the assumptions for which your answer works, it's okay not to do every possible situation. In general, we can't anticipate all possible situations. And the last one was the rate of division of E. coli as a function of temperature. And here, again, if the thing is frozen, the E. coli aren't going to divide. At low temperatures, the E. coli will divide very slowly. That's how refrigeration works. Then there's going to be a range of temperatures over which the E. coli divide pretty well. And in general, in that region, the rate of division will go up as I increase the temperature. And then you get to temperatures where the E. coli are not happy, and the rate of division will go down. Eventually, it'll go to 0 and they'll die. If I boil them, they're going to die. And so, again, you have this typical regime where for very low values, they don't behave properly. You get a region of increase for intermediate values, a saturation, a decrease, and then some kind of breaking of the system where the organisms die for high values. And so actually, the population of humans, the rate of glucose absorption, and the rate of division all have the similar patterns as a function of the control parameter, even though they're very different questions. And so I hope that homework problem was meant to be a fun problem rather than a difficult problem, just to to think about why it's not so simple always to write a simple mathematical relationship for rate. All right. And maybe we'll skip this for now, but I had a little exercise which was essentially coming back to, and this was framed a little bit differently because I had assumed that we would have a project idea already when I wrote it. But we could think about, well, let's talk about the P53 example. So what would a qualitative question be for the P53 oscillator? What could be a qualitative question? I don't have the diagram in front of me, so I don't remember what the controls are. But we could take the concentration of an external chemical species and change it. What would be a qualitative question we could ask?
We need to draw the relationship between the concentration of each component and like to the relationship. Like there are plenty of graphs we need to draw. Like if we change the amount, if we change the concentration, what would the values change? Is that the quantitative questions?
Well, that would be a quantitative question. A qualitative question would be, suppose that I have an external modulator, and I don't have the circuit in front of me, so I'm trying to remember. Maybe if Pedro's there, do you remember what the modulatory species for P50? Well, let's just take the rate of DNA damage. That one works. If the rate of DNA damage is very low, then the P53 does not oscillate. As I increase the rate of DNA damage, I go into an oscillatory phase.
Yes.
And if I increase it too much, the cell's going to die, so the oscillations will disappear. And so that's a qualitative question. In other words, I can ask as a function of the rate of damage, does P53 oscillate or not? That's a qualitative question. And I have a phase diagram, which is for low rates of DNA damage, no oscillation, intermediate damage, oscillation, high rate of damage, divergence, and death. So that would be a semi-quantitative. That would be a qualitative question. What would be a semi-quantitative question? That we could ask about this.
I'm actually confused about the semi-quantitative. What does it mean?
Semi-quantitative question is asking about the direction in which things move.
Yeah.
Yeah.
Okay.
So as an example, a semi-quantitative question could be as we change the level of the rate of DNA damage, does the amplitude of the oscillations of P53 increase or decrease?
So the semi-quantitative questions ask about the trend, and the quantitative questions ask about the numbers.
Right. So a quantitative question would be, what is the oscillation frequency of P53?
Oh yeah. Okay.
So that frequencies usually we can measure, not always, but often. But amplitudes are hard. So if you say how much P53 is there as a function of time, that's a hard one. Because generally, how would we measure that? Well, we'd measure that with a fluorescent reporter, but we don't know how many. We measure some intensity in our microscope, but we don't know how to tie that intensity level in the microscope of the fluorescence to the actual number of P53 molecules. We can see that intensity goes up and down, and so we expect that the concentration is going up and down. but we don't know how to calibrate it. We don't know exactly what the relationship between the intensity and the number is.
Mhm.
Now, there are some counterexamples to that. I mean, there are people who do things in bacteria where they actually count the individual molecules. There's a fluorescent tag on each molecule. You say there's 1, there's 2, there's 3, there's 4, there's 5. So it's sometimes you can actually count.
Mhm.
But very often you either can't count or you don't know what it means. So as an example, Pedro's doing models of a viral replication, influenza virus replication. And when you do electron microscopy, you can see the virions, you can see the viral capsids. And so you can count how many virions are produced by the cell. And you can do that with optical techniques too. The problem is that those virions don't always have the viral RNA in them, or not complete viral RNA. And so actually viruses produce somewhere between 10 and 1,000 dud viruses, where you just have the viral capsid with nothing in it or an incomplete genome in it, for everyone that's actually able to infect another cell. And so if the question you're asking is how many viral capsids are assembled, that's something that can be answered. If you're asking how many infectious virus particles are produced by the cell, that's much harder. You can only find that out by taking the virions, putting them in a dish, and seeing whether they infect or not. And so actually, viruses are quantified in a really strange way. It's called colony forming potential, which is a very operational thing. You take solution with the virions in it, you put it on cells, and you say what fraction of the cells got infected. It's actually quite a tedious experiment. It's not very accurate.
Hmm.
And so a lot of things that we think about as being apparently easy to quantify are not so easy to measure biologically. I mean, this morning I had to go to the doctor for a blood test, and they draw blood and they'll give me levels. They'll give me the level of cholesterol in my blood and things like that. Those are quantitative. But the level of cholesterol in my blood represents some average of the cholesterol level in my body. But what may matter is the cholesterol level in my liver, not the cholesterol level in my leg. And those could be different. And so that's another issue that you face, which we're not going to talk about in this class, which is spatial variability is something that we also don't have a lot of access to. Pedro, I don't know if there's anything you want to say about that as an example, but if you're there, I thought you've thought about some of these issues pretty carefully.
Yeah, it's sometimes it's hard to decide because I think for the purpose of building a model, you have to look at what the experimentalists can measure. But then you have these ensemble of papers where some of them actually can measure things quantitatively, others couldn't, but they have more information about other aspects. Some do measure quantitatively, but you don't trust the way they did. And all of those, like all of these contexts determine the decision, like if your model will focus on quantitative aspects or qualitative aspects. In general, for biology, I have trust issues with biology, so usually my decision is to go qualitative. And if it's possible to do some quantitative analysis, I open exceptions. That's more or less my approach to it.
So there aren't really right or wrong answers here. And that's really why I'm bringing this up now. When you begin to think about not just doing exercises in the homework, I mean, one thing I'm sure you've run into is that when you're a student taking a physics class or a chemistry class, and there's a homework problem, there is a right answer. There is some correct answer. But when you start doing experiments or doing modeling, more sophisticated modeling, there are certainly answers that are wrong, but often there is no answer that is definitively right. And so thinking about how to ask questions and frame questions, I think, becomes really important. If we think about our P53 example for the questions that Mogens Jensen has been working on, we mentioned that we could have oscillations or non-oscillations. And as we change the concentration of an external species, the oscillations might get bigger or smaller, or they might get faster or slower. Now, the thing Mogens was interested in was where you drive the oscillations with a periodic forcing, then you can get frequency locking, where the frequency of the oscillation shifts to be a rational ratio of the driving frequency. And actually, even in that little notebook that I presented to you the other day, we saw that. And then there was the question of how do we measure the frequency? But in that case, you can ask a question, which is, if you look at the ratio of the oscillation frequency between the drive and the response, is that ratio a rational number? Is it a simple ratio like 1 to 1, 2 to 3, 3 to 5? Or is it basically proportional? And in the case of the P53 oscillator, you get phase locking and frequency locking. So you get some quite non-trivial phase diagrams as you change the driving frequency and the amplitude of the drive. And so there are actually a whole bunch of interesting phase diagram questions that you can ask, some of which are qualitative, some of which are semi-quantitative, as well as the pure quantitative ones. So that was something I wanted to talk about, really sort of principles of modeling, thinking about problems. And now I'm going to jump to the other extreme, which is just something very technical, which is that we're typically running our antimony models inside of Python. And of course, we can use the Python to run the simulation for some amount of time, and then change the parameter of the variables in the Python layer. But it's also convenient to be able to have essentially conditionals built into our antimony simulations. And antimony has a very simple concept, which it calls an event. And that could be something like, at a given time, we add a drug to the simulation, or a nutrient to the simulation. And the syntax in antimony for an event is it can be given a name. You don't have to name it. And if you give it a name, the name has to be unique, and you end the name with a colon. And the syntax for an event then is at, parenthesis, and then some logical expression, and then a colon, and then the things that you want to have happen when the event triggers. So at time, event number 1, E1, colon, at time greater than 15, colon, A equals A plus 15. Now that actually in a procedural code wouldn't do what we want, because every time that line is called, it would add 15, so A would run away. I should also say that there is a bug, which may or may not have been fixed in the current version of antimony, actually really tellurium rather than antimony, which is that if you have two events that trigger at the same time, that write to the same value, that affect the same variable, only one of them will happen, and which one it is random. So if I say at time greater than 15, a equals a plus 15, at time greater than 15, a equals a plus 10, I should wind up with a equals a plus 25. And instead, I'll randomly get either a plus 15 or a plus 10. And I know that was not supposed to work like that. It's supposed to be fixed, but I actually haven't checked whether the latest version of Tellurium fixes that or not. Okay. So let's use a simple antimony event. If you can run Colab, you could have a simple reaction. A goes to B at a rate K1 times A. B goes to nothing at a rate K2 times B. Let A equal 10, B equals 0. I don't know why I have X0 there. That's left over from something else. I'll set K2 equal to 1, K1 equal to 0.2. And then at time 15, A equals A plus 15. And now run that and plot it. What do you get? Is that a clear little problem?
If this is not, I think that A will keep increasing.
Okay, well, can you run Colab and see if you can see what happens? Tell me what happens.
Okay.
Is A going to keep going up? In other words, is it going to go, hey, get 15, then plus 15, plus 30, plus whatever? What's going to happen when you do this? Why don't you try it and see what it does? Just take a minute, because I've got a whole series of very simple exercises. Once you have this first one done, it may take you a couple of minutes to write the first one. The changes I'm going to ask you to make each take about 1 minute each to do the remaining exercise. So take a minute to get this. And when you got it working, let me know. You can screen share it if you want.
Yeah, I posted the result
In the chat.
Okay, that looks fine. So what you found is that, which is that the value of A is increased by 15 only once. And so that conditional.
Yeah, but I
have a problem. In this line seems to be if the time is larger than 15, then A will keep increasing the number. So
Right.
I mean I think it should be at time equals 15. Then A will increase the number by 15.
Right. So that's what I want to talk about. This is what's called a trigger. So an event triggers whenever the argument of the trigger goes from false to true. And it only triggers once.
Okay.
So if you wrote at time 15, time equals 15, then do something. That will work provided the time stepper has a time when it's exactly 15. But if the time slicing skips over 15, it goes from 14.9 to 15.1, there never will be a time when it's exactly 15, and then it won't trigger.
Okay.
So you could try that. You could try at time equals 15 and see what happens. It's also true that if the argument goes false and then true again, it will happen again. And so try now adding a line at sine of time greater than 0.5, b equals b plus 0.05. What happens when you do that? And you'll notice you have to have a different name for the trigger. So E3, don't call it E1 or it won't work. So just add that single line, E3 colon at sine of time greater than 0.5. B equals B plus 0.5. What happens when you do that?
It's a oscillation like figure.
So you get repeat. Now it's repeated. It's a little hard to see because B doesn't change that much, but okay. So every time sine goes from less than 0.5 to greater than 0.5, it'll trigger. Now, in this case, we change the value of a variable. You can imagine adding the chemical to your environment at a particular time. But you're allowed to change the values of parameters too. And so why don't you try at time greater than 10, K1 equals minus K1. What happens when you do that? Get rid of that B plus, get rid of the B line. So this is reversing the direction of the reaction at time 10. I don't have any good biological reason why that would happen, but mathematically you're allowed to do it. What happens with that one?
The A
Just keep increasing.
Yeah, A will go down and then it'll start going up again. Now, if you keep running this, A will blow up, so you have to stop it. It's
Mm.
all right. Now, in this case, we've been referencing time, but your condition could be based on anything in the system that's quantitative. So you could say when P1 is less than 1, do something. Or when P2 is bigger than P1, which is a little bit more interesting. A conditional on the relationship between variables may do something pretty complicated. And so let's do a simple example of that. Why don't you try E1 at A less than 2, K1 equals minus K1. What happens? Yeah, you're going to have to run that for a little bit longer because your A didn't get down to 2 yet. So you might want to run the simulation out to time 20 or something like that.
yeah.
Okay, there you go. Good. So it goes down and it comes back up. That's what we got. Now, suppose you have a second condition, a condition E2, that says at A greater than 12, K1 equals minus K1. Why don't you try adding that and see what happens? And this time you want to make your simulation run for a longer time. Make it run for 100 time steps.
A greater than
12. Okay.
So keep the E1 at A less than 2, K1 equals minus K1.
Mm-hmm.
And now have E2 at A greater than 12, colon, K1 equals minus K1. What do you think will happen? Think about it maybe before you do it. Think about what you expect to get. So try it and then you can put the result in the chat. Yeah, so effectively now you have an oscillator. When the value of A gets to be too small, it turns around and starts increasing A. When the value of A gets too big, turns around and makes it decrease again. So with conditionals of this kind, the conditionals are not very complicated, but you can actually build some pretty elaborate relationships between things. Now, there are additional things you can do with events, and I'm not going to go into all of the details. If you go to the documentation page where it says events, you could see about additional possibilities. But that's already a lot. You could already, just with this very simple syntax, you could do quite a bit. So that's what I wanted to say about events for today. Now I'd like to come back to the Hill equation and the things we were doing last time. And we started out by writing down the Hill rate law, which had the form s to the n over k to the n plus s to the n times V max. And the key thing here is that k is the concentration, the half maximal concentration. So when s equals k, this is equal to 1/2 V max over 2. And we looked at scaled forms. There are various ways of writing this that are convenient for different purposes. But the key thing, again, if we look at this plot, is that f is always equal to 1/2 at k, when s equals k. It's always 0 at 0. It always saturates at 1 as I go to very large values of s. And as I increase the exponent, if the exponent is between 0 and 1, the function is convex at 0 and has maximal slope at 0. If n is bigger than 1, the function is concave with 0 slope at 0 and has maximal slope at s equals k. It's a sigmoid. So there are two regimes. If n is bigger than 1 or less than 1, they're different regimes. As I increase the value of n, this gets more and more step-like. You can see even for n equals 9, it's not really a step function. It's still pretty smooth. But if I made n equals 20, it's pretty close to being a square wave step function. And the symmetric decreasing Hill function, I take the numerator, instead of putting an x in, I put a k in the numerator. And then that's effectively flips around k. And so now it's 1 at 0, 0 at infinity. It's still 1/2 at k equals s. It still has maximal slope at k equals s, if n is bigger than 1. And then we talked about how to use that to interpret regulatory signaling diagrams first. And so we'll often see A activates or promotes S. That really means that A is a modulator of some reaction that produces S that we haven't just described. So we have nothing going to S. And the rate of production of S is typically, when we don't know anything else, we'll assume it when activators and inhibitors work as Hill functions. And so A promotes S, turns into nothing goes to S at a rate V max A to the n over k to the n plus A to the n. If we had S1 goes to S2, then naively we would either assume mass action, in which case our Hill function would be multiplied by V max times S. Or we might assume a Michaelis rate of reaction. V max is S1 times S1 over Km plus S1 times the Hill function. Now, K sets the concentration of A at which the reaction reaches half its maximal rate. And so if I use the same value of K for every circuit, every component by circuit, then the values of all of the variables are compatible with each other. So this is a little bit like, sorry about the squeaking here. That's a little bit like digital logic, where you have everything where a 1 is 5 volts, a 0 is 0 volts, and a 1 is 5 volts, at least in TTL logic. CMOS was different. And so here, if you have a lot of signals where the Ks are all the same, that means that the output of one can be chained to the input of another one, and they'll work together. And so that's pretty convenient. Now, biologically, that may or may not be the case. It may be the case that the different values of that different circuits do have different values of K. But if you want to build complex circuits that are going to interoperate, it's helpful to start out by setting all the k's equal to each other. Inhibition works exactly the same way, except I replace the numerator with k instead of the inhibitor R. Now, if I have gene regulation, I have to make some additional assumptions. In signaling, that's basically all I need to begin with. In gene regulation, if I say P1 is a promoter that promotes the production of gene product P2, I have to remember that this is actually something where P1 binds to a promoter site, the gene is transcribed and then translated. And when I write something like P1 inhibits or promotes P2, I always have to remember that P2 decays. And so this single lollipop then turns into nothing goes to P2, and P2 goes back to nothing. Otherwise, I'll wind up with an infinite amount of P2, which I don't want. And I'm going to assume a Hill function for the production rate and first order decay for the decay. And so V2 is minus K D2 times P2. And V1 is my Hill function. And then there's a bit of biology, which is that most genes are transcribed at what's called a basal level, even when there is no promoter. And so there's a constant leakage term, B1, which gets added to that rate of production. And so again, why would we go through all this exercise? Well, a lot of times you'll see in the literature, you'll just see P1 promotes P2. And now you want to write a mathematical model of it, you'd say, well, I don't have enough information to write a mathematical model of that. But in fact, these assumptions that we've made here work pretty well. And there actually are computer code that will take a diagram that just is the arrows and do this exact conversion that I've described here, and then run a simulation of it. Now, in reality, the values of K may be different, the values of B may be different, and so the exact numerical values are not going to be right. Values of V may be very different. However, structurally, you're going to get the right answers. This is a case where the numerical values, the quantitative behaviors of the system may not be right, but the semi-quantitative behaviors and the phase diagram should be right. All right, this is the inhibitory one. All I've done is flip the Hill function from activation to inhibition. The other thing that's pretty useful, and I realize this doesn't look very pretty when you do it, but it's very helpful, is when you have a rate of production and a constant decay, the concentration that you get out of that is Vm divided by the decay rate. And if you want to build this system so that you can have things interconnect and be compatible with each other, I want to be able to change that rate of production, that Vmax, without changing the final concentration. And what that means is that I really want to write Vmax and the decay rate as a ratio, proportionality, so that things are well behaved. And so very often you'll do that. You'll define a decay constant, KD2, which is the decay rate of the chemical. And then you'll rewrite the basal rate of metabolism and also Vmax in terms of KD2. And if I write things this way, at the bottom here, as I change KD2, I change the speed of reaction, but I don't change the equilibrium amount. And that's why you write it that way. If I write it that way, then I can change the decay rate and I can make one term in my simulation faster or slower without changing the total concentrations. That's something you may not have to worry about here. But if you're ever going to do this in practice, where you have a circuit and you say, how do I turn this circuit into an executable simulation? That's a trick that's worth remembering. The other thing that we have to do is we have to think about situations in which you have multiple signals coming in to one output. And I started that one actually early in class. I said, what if P1 affects P2 and P2 affects P3, but P1 affects P3 directly? So P3 is modulated both by P1 and P2. Now, in a signaling network, usually we have concentrations that add, and therefore the way these signals combine is fairly straightforward. In gene regulatory networks, the interaction between multiple signals can be complicated. The simplest assumption is that these are Boolean, and that we either have an AND logic or an OR logic. If we have P1 and P2, we turn on, but if we only have P1 or P2, we don't. That would be the AND. If we have P1 or P2, we turn on. If we have both, we turn on. That would be the OR. Now, we would typically want, because of the way genes are regulated, typically the rate of gene production, of gene transcription is either on or off. It doesn't care about how many regulators are bound to it. Sometimes it does, but usually it doesn't. And therefore, we have to have some way of combining these signals that's going to do that. It's also true that sometimes these things interact in complicated ways, and then you're stuck. You have to use that. So we want to know how to use these Hill functions to implement an AND or an OR. And it actually takes a little bit of thought. It's not as simple as Boolean logic. So if we want to get an AND, we want our output to be bigger than a half, if we're scaling to k equals a half. Only if x1 is bigger than a half and x2 is bigger than a half. If x1 is 1, we should get our regular Hill function for x2 and vice versa. If x1 is 0, we should get a 0 no matter what. For an OR, if either P1 or x1 or x2 are bigger than a half, we should get the output being turned on. If x1 is 0, we should get our regular Hill function. If x1 is 1, the output should be 1 no matter what. And Herbert talks about this a little bit in the textbook we've been using. His other textbook, Enzyme Kinetics, has a series of chapters on this, how this could work. For an AND function, if we multiply two independent Hill functions, so we write the Hill function for x1 and the Hill function for x2 and multiply them, then we get the AND function. And we know logically AND looks like multiplication for Boolean arithmetic. In this case, if we scan x1, this is the first function, the second one, we multiply them. If x2 is 0, whatever the value of x1 is, we get nothing. If the value of x2 is 1, we get our Hill function back. And for intermediate values of x2, we get a sigmoid where the maximal value of the sigmoid is less. Notice that the value at which we reach half of our maximum is still 1/2. It's still k. That may or may not be what we want, by the way, biologically. But this is what's called, here he talks about AND operation. You need A and B to both bind to turn on the gene. What about an OR? Now here I say and it should say OR. That's a type. For OR, we want our output to be bigger than a half if either x1 or x2 is bigger than a half. If we added the two, here we multiplied these two independent ones. If we added x1 to the n over 0.5 to the n plus x1 to the n plus x2 to the n over 0.5 to the n plus x2 to the n, that doesn't work. Because if we turn them both on, we get a value of 2, not 1. So it doesn't work the way we want it to. So here we have to think about actually biologically what's going on. We can have two situations, one of which is called competitive binding, in which case there's a single binding site, and either x1 or x2 can bind to it. And in that case, we get a Hill function where the system behaves as if the concentration of x were just x1 plus x2. And so then the functional form will be quantity x1 plus x2 to the n over 0.5 to the n plus quantity x1 plus x2 to the n. That will have the right form. In non-competitive binding, we have two separate binding sites. They ignore each other, and x1 and x2 behave independently. And so here's the competitive binding case, x1 plus x2 to the n over 0.5 to the n plus x1, x2 to the n. Here we get a sigmoid in x1, and as I increase x2, the sigmoid gets shifted to the left. That's a very different result from the one we had for the AND, where the amplitude varied, but the switching position didn't vary. So in this case, the critical concentration gets shifted to the left, but the amplitude doesn't change. For non-competitive binding, x1 and x2 are independent. We have x1 to the n plus x2 to the n divided by 0.5 to the n plus x1 to the n plus x2 to the n. In this case, if x2 were 0, I just recover my Hill function for x1. If x1 and x2 are both big, I get a value of 1, which is what I want. But this one looks like the AND case. In this case, the switch is still at a half, always. It's not shifted left and right. But the amplitude of the response depends on the strength of the other one. And so this is essentially exactly the AND case, but flipped upside down. And so here we have two, this should say OR rather than AND, so I have the same typo. In this case, I have two very different kinds of OR gates, and they're going to behave biologically very differently. And so I have to make a decision about which one is biologically meaningful or not. And that's not something that I can read off from the diagram. That's something that I have to try. I have to try both. If we have signals that have different k values, then we have to rescale everything to be with respect to the k. And so things become a little bit more complicated. And so if P1 has a Hill constant of K1, its range will be between 0 and 2K1. And so to make it possible to combine things, we have to convert them to something between range 0 to 1. So we divide by 2K. if we divide each one of our variables by 2 times its Hill function, its half maximal value, then each one of our variables will range between 0 and 1, and then we can build circuits that work together. And so the process here is a little bit more complicated. We say we have P1 and its corresponding K, P2 and its corresponding K. If we want to combine them, we divide P1 by 2 times K, P2 by 2 times K, get X1 and X2. Now we do the combination, then we plug in again and get it back out. And so if we want to combine P1 and P2, we get these scaled forms like this. And this allows us to combine things that don't have the same value of K. Okay. So again, we did this in the simple case last time. So now let's go through that exercise. And I think you did this in the homework, but maybe we can do it together again. Because I think there may be things that are worth discussing. If you've got your homework problem, you can just pull it up and we can look at it together. Here we have A promotes itself. A promotes B. B inhibits A. Where is there a combined regulatory circuit? And here I wrote the slide assuming there's more than one person in the room, but you've done the homework problem, so you've thought this one through. So B only depends on the concentration of A, so there's no combination here. But A is promoted by A and it's inhibited by B. And so I have to decide how that incoherent signal of A and B combine. And there's a general trick, which is that when you have coherent signals, A and B both excite something, or A and B both inhibit something, then you can have, typically you have an OR combination. You have an inhibition combined with an activation, usually the inhibitor wins, and so you have an AND. So the simplest thing here would be to assume that the signal is multiplicative, that you have an activating Hill function for A multiplied by an inhibitory Hill function for B. Although Herbert actually comes up with a rather complex functional form for this one, and I'm not exactly sure where he gets that. So I don't know if you wrote the rate equations for A and B using our Hill function formulas. When you did this, how did you do it? Did you go through the procedure that we had? Did you look it up? How did you come up with equations for A and B?
I need to have a look. I couldn't remember my answers.
Okay. So maybe it's worth trying to do it again, not looking, just based on what we just did and the rules that we just came up with. Try doing it again and see if you get the same equations. So the way I would recommend doing this is starting out writing just A. A activates A. And we just did that one. We have the equations here for that. So you could pull that. And then A activates B will do the same thing. So you can write the equation for that. And then you can write the form for B inhibits A, the Hill function. And then the last thing that you're left with is how do I combine the activation and inhibition of A together in the rate equation for A. So why don't you just give it a minute or two to think about that, see if you can come up with equations that make sense. And I should say that, I mean, I have ones that I used, but there are multiple equations that you could write for this that would be reasonable. What did you get?
I'm not sure. I just wrote every separate. reactions. Like there are 5 in total, and I'm trying to add all the V's, the speed.
Okay. So here's what I came up with. So if you wrote it independently, the activation would be K1 a to the h over 1. Here I used 1 instead of a half. 1 plus a to the h. That would be the activating function. The inhibitory function would be K1 times 1 over 1 to the h plus b to the h. That's the inhibition. The B1 is easy. It's just a to the h over a to the h. And so here what happens? If A is big, this is turned on. And if B is big, it's turned off no matter what A is. In this case in particular, what matters is whether A is bigger than or less than B. If B is bigger than A, it turns it off. And if A is bigger than B, it turns it on. And so this is actually triggered by the relative values of A and B. So this is and like rather than or like. You could try other versions and you'll see it should still oscillate, but the oscillations will be a little bit different. But in general, again, the rule is that if you have an inhibitor, the inhibitor wins. And I realize that that's not always a trivial thing to realize. Okay. So given this, why don't we use these equations? Why don't you try plotting the time series for it? I think you had it. I don't know whether these are the equations you used or you used a different set. But why don't you show me what the time series look like for this?
I think I did it in the board.
Okay. Do you want to put the results in the chat? Maybe.
Yeah.
And you'll notice that the values of K are rather strange. These are ones Herbert picked specifically from a published paper. I normally try to make things so that they work when the Ks are all about 1. Clearly, that's not the case. Particularly the lifetimes of B is pretty long, so K3 is small. Let's take a look. So in this case, what you have is that
The first picture is
Using the parameters you gave here.
okay, so for the first one, it reaches a steady state.
Yes.
Okay, and now I said write a parameter sweep for A naught and try it for different values of B naught.
Yeah.
And what you get here is that for some of those parameters, maybe you could screen share this and we can talk it through together, because that may actually be, it'd be good to look at that. The thing you just put in the chat, why don't you screen share and let's look at that together?
Yeah, sure. I can share the screen.
Because it's sort of an interesting set of results when you think about it. There are only 2 variables, A and B.
It's here.
Yeah, there we go. And in this one, so the only difference in the left-hand one is the initial values of A and B. Is that right? They all have the same values. They all have the same Ks and everything else.
yes.
And so one of them goes to a steady state and the other one oscillates. And so instead of
Oh yes, right.
And so instead of having a choice between 2 fixed points, you have a choice between a fixed point and an oscillator. That's a little bit unusual. It's not always, that's not something you see that often. And what's the difference between the one on the left and the one on the right?
Left is A, the right
Is B. Is
So on the second one, it's still the same parameters, but you're scanning different set of initial conditions. Is that right?
Yes, yes.
Now, one thing I'm not sure of with the second one is if you ran that for a long time, are those oscillations going to persist or they're going to disappear?
I haven't tried yet, but I think it will disappear eventually. Like the amplitude is decreasing.
So why don't you try that? Why don't if you have that code, why don't you try running that for 10,000 time steps instead of 1,000 time steps? Let's see.
Sure. So it should be.
Now, something that in principle we'd like to do would be to do a phase diagram here. It looks like it doesn't, it looks like they're persistent, huh? You're probably going to have to increase the number of sub-steps. When you go for a long time, you're probably going to have to increase the number of where you do where you call simulate. I want to make the number of points bigger, yeah.
5,000.
Yeah. Because if the step size is too big, you can get numerical instability. So, yeah, there you go. See there you don't get that scalloping in the oscillation.
Yeah.
So you do reach a steady state. You do reach a stable oscillation.
Mm-hmm.
So that's a sort of an interesting result. So you can have a high steady value, a low steady value, or an oscillatory value.
Mm.
So the bifurcation diagram is fairly complicated here. So let's and here's some possibilities I've got some different ones. So you can reach a steady state or one where it oscillates around a small value. And now we can do the phase portrait. And the phase portrait is sort of interesting. Why don't you, did you do this one?
The phase portrait. Oh,
Okay, so you have the code to do the phase portrait. I know, I saw that you have it. So why don't you try to put this inside of a phase portrait? So now plot the trajectories for different initial values of A and B.
Okay.
And let's see what happens. Because this one is actually rather fun. It's quite interesting. And it is going to bring up this idea of null lines, which we haven't really talked about yet, but it's a good motivation. Pedro, did you do this particular oscillator? So it's actually a more complicated oscillator than I would have thought, given how simple the circuit is.
Yes, in the 1 year ago when I had this course, yes.
It's a very peculiar oscillator. We're used to sort of, it's not a simple hop bifurcation. It's sort of a complicated. And I have this comment here that when you try to do the phase portrait using the code that I provided for you, or Herbert provided for you, yeah, there we go. You may actually wind up having to do 2 plots because the position, those oscillations can be pretty small. And so if you plot so you can see the oscillations and you can't see the steady state. So you may actually want to do 2 diagrams with different ranges of A and B. A way to figure out how to do what the ranges are. Let's look at this picture. Did I? No, I killed the screen share, didn't I? So let's come back to the screen share for a second. You'll notice if I look at the plot that the high steady state has a value of about 40. And the oscillators go between a value of about 0 and about 5. And so if I look at, if I do a phase plot with a range between 0 and 10, I'll see the oscillators. But to see that second steady state, I have to go out to 50. And so you may want to actually do two simulations, one of which you scan the values of A and B between 0 and 10, and the other one of which you scan the values of A and B between 0 and 50. Otherwise, it's a little bit hard to see what's going on. That's an easy thing to change in your simulation. And so, did you get something that looked a little bit like this? I think if I look at your picture is the one on the left.
Yes.
Okay, so what are these red lines? These red lines are null lines. And what is a null line? A null line is simply the places where the flow is either horizontal or vertical. And probably I should have used a different color. So if I look at the null line on the left, you'll notice that the flow is horizontal whenever wherever it crosses the red line, the left line, the left red line. And so the flow goes from increasing value of y to decreasing value of y. And here y represents b. So this is where dB by dt is 0. The flow is horizontal. The second one, the one on the right, represents the places where the flow is vertical, where dA by dt is 0. And the reason you use these diagrams is because within a quadrant specified by the two null lines, within this box, can you see my cursor?
Yes, I can.
So within this box where my cursor is, B is always increasing and A is always decreasing in that box. So the flow is always up and to the left. Below that line here, in this bottom right-hand region, A is always increasing and B is increasing. So the flow is always up and to the right. The left-handmost box, wherever I am, A is decreasing and B is decreasing. So the flow is always down and to the left. Now, there are these small regions here at the bottom, which are a little bit more complicated to analyze. But within any given box, the flow is always in one of four directions. It's either up left, up right, down left, or down right. And so the overall qualitative behaviors of the flow are uniform within one of these regions. That's why you draw these boxes. You'll notice wherever two null lines cross, you have a fixed point. You have a fixed point when the derivative of both A and B is 0, and that's precisely where they cross. You'll notice they cross twice. There's this green dot basically along the x-axis. And then there's a second dot, which is red, which is here at a value of about 0.8, 1.5. The green dot is what's called an unstable fixed point. If I move a little bit away from that green dot, my flow is always away from the green dot. So if I start at the green dot, I stay there. But if I move a little bit away, I go far away. What about this red dot? This red dot is an oscillatory fixed point. If I start exactly at the red dot, I stay at the red dot. If I move a little bit away, I oscillate around it, and I can see my flow. Here, A increases. I cross this line, and B increases. I cross this line, B is still increasing, but A is now decreasing. I cross this line, A is still decreasing, but B is now decreasing. I cross this line, B is decreasing and A is increasing. That's my cycle. So I go around and around and around this point. It's not circular. It's got this elongated shape, but that's my oscillation. Notice something else happens. There's something we haven't drawn called a separatrix. If I start with a value of B equals 0, X equals 6, what happens? Where do I go? I go off and to the right. B increases and A both increase. What if I started with A equal to 2 and B equal to 3? I'm still in the same region, but in this case, I get swept into this loop around that fixed point. And so there are two different destinations within this region. So the null line defines the places that the direction of the flow changes, but I can have multiple outcomes. I can have multiple fixed points within a given box of my null line. And again, don't worry about this too much. We'll come back to it. And have you studied, you said you'd done some dynamical systems or not? Had you ever had a class in dynamical systems?
No.
No. Okay. So in that case, I mean, these are things that would be true of the oscillators you studied in physics, but they may not have been framed in exactly this way. Now let's look on the right-hand side. This is exactly the same plot, but on a bigger scale. Notice that the x-axis now goes between 0 and 20 instead of between 0 and 7. All of those oscillator loops are now very hard to see because they're all squunched down in the left-hand corner. But we see a second fixed point, this green one, where the two null lines cross. If we start for very small values we start with a 0 value of B and a value of A of, say, 12, I go off and to the right, and I get lost. This is called, this fixed point here, the green one, is called a saddle. If I start up above it, I move down, and depending on whether I'm on the left-hand side or the right-hand side, if I'm on the left-hand side, my value of A gets swept to being small. If I'm on the right-hand side, my value of A gets swept to be big. If I start up at the top and to the left, I get swept down, I come all the way down, and then I start circling around that oscillator. And now what I really should do, and I don't have it here, I don't think, I don't have it here, is can you redo this? Can you do the plot now? Where instead of going from 0 to 20, we go from 0 to 50. Because there's one more fixed point. The things don't go to infinity. Look, this looks like things are going to run away to infinity, but they don't. And so there's actually one more of these that we should have done for a bigger range. And so we've seen the fixed point along the y equals 0 axis. We've seen the oscillatory fixed point at 0.7, 1.2. We've seen the saddle fixed point at 10, 12.5. And there's going to be one more fixed point. Let's find that. And that'll be enough for today. And then next week, when we come back to this, I'll explain to you the mathematics and how to analyze this. I first wanted you to see what this looked like and play with it and begin to develop a little bit of a sense of here we go. Yes. So let me screen share the picture that you just sent me. And can you explain to me what you see happening here?
So the fixed point at the near a equals 15.
Yeah, that's right. And that one is a stable fixed point. All of the lines come into that and stop there. And so what happens here is if I start out small, I'm going to cycle around that fixed point all the way on the left for small values of A. For large values of A, I'm going to wind up with that steady state around 50. And if I look at what happens, it's not so simple. It's not so easy to tell what's going to happen for a random initial value of variables. If I look, for example, for a large initial value of B, where is the threshold that determines whether I oscillate or go to that stable fixed point? If I start with a big value of B, where is it? If A is bigger than about 15, I go to the big value. If A is less than 15, I go to the oscillator. What's a bit surprising is for small initial values of B, I go to that fixed point, right? If I start with an initial value of B of 1, and I start with an initial value of A of 2, I'm going to go to the big fixed point. So it's actually hard to get to the oscillator if my initial value of B is small. And so now I could color this, and this is something you could try doing. I don't have the code to do it, but you could color this now based on what are called the bases of attraction. You could run through each point and ask the question, for each initial value of A and B, do I wind up cycling around that fixed point on the left? Do I get an oscillatory result? Or do I go to that fixed point on the right, the one about 45, 10? And that'll define the basins of attraction. And you can see where they are. And the boundaries between those are called separatrixes. And they're related to, but not the same as the null lines. And so now we're seeing, even for a very simple system, we have two variables only, A and B. We have rather a complicated set of behaviors already for this very simple circuit. And we've been introduced to the idea of null lines a little bit, the ideas of basins of attraction, the idea of oscillators versus fixed points. And then you can play some interesting games, because you can have a situation where you're in an oscillatory state, you kick it, and it jumps to the steady state. The steady state, because it's such a big value, it's hard to kick it back, but it's easy to kill the oscillators. Okay, so we're out of time for today. Was there anything else you want to talk about? I realize that we've been exploring this. Mainly in a very qualitative way. But I hope this motivates you to start. You could read a little bit about separatrixes and dynamical systems. Herbert has pretty good that chapter 13 where he talks about stability is pretty good about this analysis, but he doesn't actually do this one out in detail, which is why I thought it would be interesting to do it. Go ahead.
Oh, I was about to ask about the project.
Okay, go
Yes.
ahead. Please do.
So the postdoc, sorry, I forgot his name.
Hayden, yes.
Oh, could you ask him to send me some descriptions via email about what am I required to do specifically? We can have a discussion.
I will send you the project assignment.
Okay, great.
I've been holding off in previous years. I sent out the project assignment at the very beginning of class.
Mhm.
Two things happened. One, it scared people away, which wasn't so much of an issue here because people thought it sounded too intense.
Mhm.
And two, people weren't ready to start thinking about it because they didn't know enough about what kinds of things they were interested in.
Mm.
And so I wanted to get us through a little bit of this before we started about the project. But what I would like to do, and then I'll send the assignment. And the assignment's a little bit formal. We don't have to do exactly what's in the assignment.
Oh, okay.
But what I'd like to do is have essentially a tutorial. And I think if it's okay with you, I think this P53 one is pretty nice.
Mm-hmm.
And so what I'd like to do is you present the biology in a few
Okay.
paragraphs. You present the mathematics in a few paragraphs.
Mm-hmm.
Then you have code that will run the time series.
Mm-hmm.
And you can show, gee, if I change this parameter, I get different frequency locking. If I change and then you have code that'll generate the phase diagrams, that'll generate the flows.
Mm-hmm.
And so somebody can learn about all the really interesting things that happen in that circuit, starting with the biology and the mathematics, and then you give them some sliders so they can change parameter values when they run it.
Mm-hmm.
So the goal is that somebody who reads your write-up of this can understand why it's an interesting circuit, why it's important biologically, and also why it's interesting mathematically.
Mm-hmm.
And that's why these examples that Hayden was showing, I think, are useful. They're not from this class. In other words, they're from a class that's being taught at Purdue.
Mm-hmm.
But I think the style or those ones that we had on the NanoHub ones were quite good. I asked you to look at 2 NanoHub examples.
Mm-hmm.
Those are pretty good. The difference is NanoHub is limited in terms of what you can do in terms of sharing, although we could put it up on NanoHub if you wanted to. The thing about putting it on NanoHub is you actually get a DOI of your own. And it's not very different. It's a regular Jupyter notebook. So if it runs in NanoHub, it'll run in Colab.
I think there are plenty of scripts that focuses on the P53 on NanoHub.
If you found one that you like, then maybe we don't need to do it again. But I don't remember doing the frequency locking on P53. I did have a student who did the P53 oscillator a few years ago, but they didn't try driving the oscillator with a periodic forcing and see the frequency locking.
I see.
So that was that's what I thought was really interesting about what Moens did.
Okay. I thought it has already been well studied.
Well, I mean, I'm not expecting an original piece of research for this class. You're welcome to do it if you want to. What I'm expecting from the class is that you digest a number of articles,
Mm-hmm.
You understand them well,
Mm-hmm.
and that you can present them in a way that teaches other people about it.
Okay.
That you have your own, your ideas are not so much in the basic science, which we're bringing from somewhere else, but rather in the way you explain it to somebody who doesn't know it.
Okay.
Because if you look at Mohn's papers, they're not so easy to read if you're a beginner.
Yeah.
And so, I mean, again, sometimes people do original research, but this course is an introductory course. So, the expectation is not that this is your PhD. The expectation is that you're learning about techniques and learning how to explain.
Okay. Okay.
Okay. So I'm not going to be, I mean, I do expect a nice job. In other words, I expect a notebook that is clear, that's in your own words. It's not just copied from Mohn's words. And that really teaches somebody who uses it something new about how this mathematics works and the biology works.
Okay.
But I'm not expecting you to come up with, gee, there's a whole new oscillator in P53 that no one ever thought about. No, that's unreasonable.
Okay.
Okay, does that help? And also,
Yeah.
let me review the assignment as I wrote it last year tonight, and then tomorrow I'll send it to you tomorrow.
The homework 5.
Well, no, this will be the project assignment.
Oh, okay.
Because again, the project assignment was written assuming that there were a lot of people and that people would work in teams.
Okay. Okay.
And so I have to change it a little bit so it's appropriate.
Mhm.
But again, the goal here is to have something which is fun and where you learn something, and where what you write is something that other people can learn from. In other words, if you think about it this way, suppose you were going to write a lecture for this course on the P53 oscillator. And instead of putting it into a PowerPoint deck, you're going to put it into a Jupyter notebook.
Okay.
And so there'll be exercises for the person to do, and they'll explore it, and you'll explain frequency locking, period doubling, and these kinds of things. Because all of that happens. That's why it's such a great problem. You've got all these fun periodic, all these fun oscillations and things that happen in this system. And again, if you really want to do a different circuit, we can do it. But I think the P53 one, it's really pretty and it's not so difficult.
That's interesting.
Yeah. Okay. Okay. Is that anything else you want to talk about for tonight?
No.
Okay, great. Well, thank you always for being patient. I really appreciate you. I know it's not easy being lectured at when you're one person, so I do appreciate being there. Okay. I'll see you next week then, and I'll try to get that deck to you, the assignment to you tomorrow.
Okay. Thank you.
Thank you. Good night.